good people don't smoke marijuana.
I've known Larry for a long time.
Quiet, numbskulls, I'm broadcasting.
This is the Medical Marijuana Radio Show with Larry Love.
Hello, everybody. Welcome to another edition of Medical Marijuana Radio. This week we have Dr. Anna Schwabe, a cannabis geneticist and researcher, and we're going to get right to it. Here is Anna Schwabe. And we're back, and today I have Dr. Anna Schwabe on. She's a cannabis geneticist, and we're going to talk all about genetics and what she has found out in her research. So welcome to Medical Marijuana Radio.
Hi, great
Hi.
to be here. Thanks for having me.
So tell me about your passion in getting into the genetics of cannabis and how that came about.
So my background is in cellular and molecular biology, also anatomy and physiology. And then I did my master's degree in conservation genetics, looking at rare plant species. And then following that, I worked at the Denver Botanic Gardens. And while I was there, I had a volunteer in my lab who was in the cannabis industry, and we'd have conversations about various different things. And some of the things that we discussed, I thought that it would be really easy to clear up some of these questions. And so I had this idea. The first idea was to look at the same strains, but purchased from different dispensaries, because I had a lot of people saying, yeah, it's different everywhere you go. And I thought that was just not okay. Strange, you know, when you buy a product,
Mm-hmm.
it should be the same wherever you get it from. And so I approached my mentor that I had for my master's degree and asked him if I could do this project. Of course, he said no, because cannabis is federally illegal. And but he approached the school, and the school decided, yes, it was okay for me to do this project. And my original project, of course, I did that, but the rest of the project just sort of evolved as I was going through my PhD, and I think we did some really good work, and it was super fun. And now I'm here.
Uh-huh. So how does one like map if that's the right word, because I'm not a scientist or anything, map the genetics of a particular strain? Okay, because you said that it was different from everywhere. There's a lot of reasons that could be. So how do you map a strain to see if those two are actually the same?
So the genetic tool that I use, it doesn't actually look at the genes that are being expressed or the genes that are present. What the tool that I use in my genetic work is, they're called microsatellites, or short tandem repeats. And in the non-coding portions of the genome, there are, in every living organism, there are sections where you have little repeating sections over and over again, that vary among individuals the further apart they get in identity. So presumably, if you're closely related to something, you're going to have that repeating unit will be the same. The further you get, that repeating unit will change over time. So that's and it's commonly used in all kinds of plant identification. So like tomatoes, apples, olives, grapes, all cultivars usually have some sort of microsatellite region that can identify this as a unique cultivar. And for what I was looking at, you know, if I buy Durban poison at dispensary A and Durban poison at dispensary B, ultimately those should be identical to each other.
Not in every way, though, right? I mean, as a novice end user and someone that's been studying and talking to people for the last, I've been using the plant for 50 years, but, the last 20, it's been, getting into the science of it, which has been very fascinating. They could be different in like the strength of the particular terpenes that are in there and flavonoids.
Oh, I didn't look at any of that stuff. I mean, to start with, I didn't look at any of that stuff. I just took the genotype. I just took the DNA and compared. Is this the same as this? It should be, because whether you have clones or whether you have feminized seed, or even, seeds from the same cross, they should all be very similar to, clones should be identical, right? So Blue Dream, for example, was back then a clone only strain, because when you do the feminized seed, whatever, it would come out, you get different phenotypes and things like that. So clone only strains, they got to be identical. You're cutting things off the mother, growing out a clone. So there should be no genetic difference in the core DNA of those individuals.
I see. So there are like some like markers in there that will always be there if it's from the same strain, is what you're saying.
They
Okay.
should be, yeah, and you should very easily be able to identify. And there with feminized seeds and things like that, you're going to have a little bit of variation, and we did expect to see a little bit of variation. But honestly, out of the 30 strains that I tested, in 27 of those, there was one seriously different genetic outlier that is clearly not anything similar to the other samples that we had in the set, which tells me that there's something going on there, and we can't pinpoint whether it's who's selling it, who's growing it. We can't tell if it's been mislabeled or relabeled for a quick sale. We don't know.
Right.
But the point is bringing education to the customer that, yeah, if you think that your medicine differs between these two dispensaries, they probably do. And it's probably not just due to environmental conditions, which can also have a very big impact on your final product. But if you don't start with the same genotype, you're not going to end up with, there's no chance in ending up with the same phenotype.
So can you take a cross, let's say you cross that Blue Dream with something else to make, a new cultivar. Can you see the same Blue Dream markers in that F1 of a new cross? Is it carrying through that you can see scientifically?
Yeah. So yes, you can. So for example, I have things like Island Sweet Skunk and Chem Dog and Durban Poison and some of those things. So I had 10 different markers, so 20 different alleles that I was looking at. And you kind of take all of that information, and there are statistical programs, there are programs you can run it through that will tell you how closely related these things are, which will also bring out relatedness statistics. So 0 being not at all the same, like not even related, 50 or 0.5 being, let's say like aunt to niece. You've got siblings, which would be, 0.25, and then you've got, or I'm going the wrong way, 0.75, and then you've got 1 would be a clone. So if you're, 1.0 relatedness, then that means you're identical. So you can look at those relatedness statistics, and you can and I did see some of these things where I looked up, where did, these certain strains come from? Who were their parents? And some of them you could totally tell. It's like, yep, those two are 50% related. That makes sense.
So can you tell that Blue Dream has the markers from Blueberry and Super Silver Haze? Can you see that going?
I didn't have Blueberry or Super Silver Haze in my sample set. So you would have to have, all of the things in your database, in your numbers, to be able to make comparisons like that. But, generally speaking, and I had no funding for my PhD. It was all little bits here and there and a lot of out-of-pocket expenses. All the cannabis that I purchased, I bought out of pocket. And it was really just what was on the shelves. I didn't tell anybody what I was doing. I didn't, announce that I was going to put this into a research experiment. I just went at it as a consumer would. I looked things up on Leafly, and certain things sort of, what do I want to say, just came out in the result, like that I didn't know going in. For example, Chem Dog, I didn't know anything about Chem Dog, but going around to different dispensaries, I found Chem Dog D, Chem Dog 91, Chem Dog 4. I found Chem DOG and Chem DAWG. And I was like, okay, I don't know what's going on here, but they all seem to be some sort of Chem Dog variant. So I bought all the Chem Dogs I could, not knowing anything about them, and then found out about the legend of Chem Dog and the bag seed and all that. And the variation that went on in the Chem Dog samples that I have was actually super surprising, and it was totally exactly what you would expect to see from bag seed.
So can you tell me about the relationship between those different Chem Dogs? Did you see?
There was a little bit of variation, but they weren't clones, but they also weren't super variable in their genetic makeup from each other, which tells me that these guys were all related. All the samples that I had were all related in sort of a sibling fashion, more than, a clonal relationship or like distant relatives or something like that. So
Is it easy to see different phenotypes of the same cultivar in the markings? Like if you have an F1 of across, they all could show different expressions, until you stabilize it, right?
yes. So like I said, with the markers that I use, they're not actually in the genes. They're in the non-coding regions. So you're
Okay.
not going to know anything about the phenotype from the markers that I used, where I was just looking at relatedness
Hmm.
and how, if they're identical to each other or not. That was the question. Are they identical? Yes. Or are they identical? No.
Yep.
But yes, you can have clones and you can grow them in different places. And yes, they will be very different.
Right.
So,
And what you
follow-
feed them as well, right? How
it's way more than that. So, second portion to my PhD work, I was looking for sources of variation. So I went around to various dispensaries, bought, the same thing. So I had, I think, 3 Durban poisons. I think I had 4 Blue Dreams, some Ma Boss, and some OG Kush. And I tested them all and found the ones that were genetically identical. And then I took those to a lab and had the cannabinoids and the terpenes measured because they were genetically identical, at least from my markers. And I wanted to know what was the difference between the terpenes and the cannabinoid levels, and they're all completely different. So because they're from different dispensaries, and I'm talking not like, a couple of dispensaries on Broadway in Denver. I'm talking about Greeley, Fort Collins, Denver, Breckenridge. So presumably they were all grown in different facilities. And the things that feed into that phenotype difference is not just, the nutrients. It's water, temperature, light, nutrients. It's cultivation time. So were they harvested at the exact ripeness? Were they cultivated early or late? The curing process, the time to sale, the storage conditions are all going to change that chemotype, even if you start out with the exact same genetic identity.
You're talking about clones, and you mentioned that. So, have you done any research in regard to when you use a mother too long, it sort of degrades, and you're not getting, the clone exactly like you did maybe a year ago? And can you tell me anything about that?
I can, and I have a theory, and I did some work before I left at my last couple of semesters during my PhD, but it's still sitting. It's still sitting on the shelf. I haven't collected or analyzed that data yet. But my theory is that cannabis is very prone to changing under stress, and cutting a cannabis plant, taking a bunch of branches off, is a stressor, not only to the mother, but also to those clones that are cut off. Now, that process of cloning is not going to change inherently the genome, the DNA sequence in those individuals. But what can happen is epigenetic markers can be added as sort of a stress response, meaning that marks can be added like methyl groups or the histone tails, which is just another part of what the DNA is wrapped around in the cell. But things can be added that will change expression, either make it more difficult for that gene to be expressed because something's now in the way, or it can make it easier for some gene to be expressed because now it's opened up and can't wind back up correctly. This is all like, I'm trying to explain it the best way I can, but it's going to change the expression because the plant sees stress as, oh my God, my life's ending. At least some of them do. Oh my God, my life's ending. I need to do something to try and survive. And it's not like a conscious decision. Oh, I need to do this. It's more like a Hail Mary. Like I'll just and try to
Just
survive.
Putting out stuff, whether it's pest management for itself or anything else.
Yep, exactly. End of life. And that's why a lot of plants under stressful conditions, if you stress them, they're going to throw out a banana or, a pollen producing structure because it needs to make a seed to make sure that gets its genes through to the next generation.
Right.
And it'll make seeds. So, and some plants do that. If you drop a nickel next to them, and some plants will just not do it at all. Like some plants are very tolerant of stress and some are not at all. So the level of stress will kind of dictate whether that plant will be able to stay stable over many, many generations of cloning, or there are lots of plants that you cut them a few times and they're just done. Like the clones are not the same. The mother is kind of just not the same. And it's not always bad. Sometimes it's just a change, right? It's not always something bad that happens. Sometimes the terpene profile just changes, but it's not the traits that you were looking for. It's changed. So a lot of times people will just be like, well, that's no good anymore. So my theory is that it's epigenetic changes, which can be passed on to next generations for several generations. And so what I did, which is the data that I haven't analyzed, is I took clones from a mother and then took leaves from before and after, and then cloned those clones and then cloned those clones of clones. And I did, I think, 6 or 7 generations of cloning. And eventually, hopefully, I'll get around to looking at just the methylation in the genomes to see if it changed. I'm not looking for, is there more methylation or less methylation? I don't have an answer to that question. But did it change? And if it changed, then that tells me that, yes, there's something going on in the epigenome that could be leading to what people see over time in their mothers or their clones kind of changing.
Can you look at markers on some cultivars and tell by the markers if they're sativa or indica or ruderalis? Let's throw that in as well.
So ruderalis is a mutation in the flowering time. That's really what that is. It's not like a different species. It's just a mutation in the flowering time, and a mutation doesn't mean that it's something different or different enough to call it like a new species or subspecies. If that were true, then freak show would be a new species because it's got a mutation in the flowering or the leaf shape, right?
I was just about to ask you about freak show.
So a mutation is not necessarily something that you would say, okay, this is something totally different. It's not completely different. However, getting at your indica sativa, it depends on what you're talking about as far as indica and sativa. Are you talking about the growth pattern? Like the indicas are short in stature and they have denser buds and darker in color, and sativas are taller and spindlier and fluffier buds. Or are you talking about the effects? Is it an indica type effect or a sativa type effect? Because those two things have zero to do with each other. You cannot tell by looking at a plant how it's going to affect you. Yeah. So when you ask somebody if you can tell genetically if it's going to be an indica or sativa, there is no answer to that because I don't know what you're asking. Are you asking about effects or are you asking about morphology? Right.
Well, they're starting to use wide leaf and thin leaf varieties now, because there's been so many.
Okay, so I know exactly which paper you're referring to, the broad and narrow leaf variety. That again is a morphological character and it has nothing to do with effects. So it really doesn't matter, what you just have to be very specific. Are we talking about morphology or are we talking about effect?
As far as
scientists have tried to mash these two together and they've said there's no such thing as indica and sativa. That's not true. There is something genetically inherent about, growth patterns. Or it could be rapid local adaptation, also known as phenotypic plasticity, or geographical region, local adaptation,
terroir, Appalachia, all that stuff.
Yeah, and yes, exactly. And then as far as effects go, when you look at those morphological characteristics, and then there's nothing holding the indica type of, people say this is an indica, and we know it grows like this, and they're trying to match these two together. They don't match together. They're not correlated in any way. But I do feel like there is something in the terpenes or the cannabinoids, and I don't think it's anything that we found yet. Keeping in mind that terpenes and cannabinoids can pack a big punch while still being in only trace amounts. And so we haven't looked at the whole we only look at a handful of each, right? So I think there is something to the described effects, and everybody's effects are personal.
Yes, that's what I was just about to say. What throws a big wrench into deciding what the effects are is that our human bodies are affected by cannabis in different ways. The same, a person may come in. I always use this example. When I worked at a dispensary years ago, someone would come in and they would say, the AK-47 was really great for me last time I got it. Can I get some more of that? And then the next person would come in and go, listen, I didn't like that AK-47. What else do you have? So that sort of messes the effect thing up because it just affects us so differently.
But it doesn't, though. If you think about medicine or anything really, what I like and what you like, cannabis or not, is incredibly personal. I might like wine. You might like beer. And I can't tell you, oh, if you don't drink beer, if you drink beer, you're going to get tired, because that's how it affects me. You're going to, it's different. Same thing with medicine, just regular old, pharmaceutical medicine. You don't go to a doctor and say, I'm depressed, and they go, oh, here's exactly what you need. They're going to try a medicine, and then you're going to come back and be like, oh, I didn't like that. I couldn't sleep. I broke out in hives. Can we try something? And they'll write you a different prescription. And then you've got dosage adjustments and things like that. So it's really not this indica sativa thing that's the issue. It's saying indica will make you this and sativa will make you that, because that's the problem. You can have categorical groups and just say, this is an indica, this is a sativa, try both, see what you like, and or not. You might like one during the day and one at night. See how it affects you, see what you like, and then come back. And then we can start to hone in on, your personal needs, requirements, desires, wants, whatever the case may be.
Right. Well, one of the problems is consistency with the end product. Okay. So you have to have trust in where you're getting your medicine from. They grow in different ways. When we're talking about medicinal, obviously, we now have proliferated, adult use, and that's changing the game a little bit. And I think a lot of the medicinal products are disappearing from the shelves. I've been talking about that. But the fact that we all react differently to it is one of the hardest parts of people not having a bad experience. If you have a bad experience the first time you use it as a medicine, you're not going to go back to it.
And I also feel like because we attach effects to these names, if you tell somebody, here's a sativa, it will make you feel like this, and then they go home and they try it and they go, I didn't feel like that at all. Like this was horrible, a horrible experience. They're going to think there's something wrong with them, and they may be really turned off because they didn't have the experience that you told them that they were going to have. And so I feel like we need to get rid of this whole, this will make you feel like this, or this will make, or people, like Joe Henry down the street uses this for his migraines, so it's also going to help you with your migraines, because that's not true. Like Joe, like people have to experience these things for themselves and kind of make some decisions based on their personal experiences to get them where they need to be. And a lot of that is trial and error, but that is the same with any kind of medicine. But I just did a conference. I just did a presentation on this, and it was at a pharmaceutical conference. They wanted to know how do we decrease variation in medical cannabis. And first it starts with genetics. You got to start with the same thing, so that everything's on a level playing field, and then we can address the other things that lead into variation, which are the environmental conditions, including air, light, temperature, all that stuff, nutrients, water, and when you harvest, how you cure it, how long it stays in, or how you store it, and age to sale. And that is the only way that you're going to decrease variation in cannabis. So I am a proponent, at least for medical cannabis, of standardized growing conditions that everybody across the country and across the world.
I'm sorry, I'm laughing because growers have such egos.
No, but wait.
Yeah, okay.
But no, this is just for medical cannabis.
Okay.
And so everybody has this idea of how they need to grow it in order to get this product. However, yes, growers are awesome. They all like to put their own spin on it. They've got their nutrient regimes. They know how to produce the best product for them. And from that, you would have to stray from these, standardized growing conditions that are very regimented and blah, blah, blah. And I don't think that's an issue. You just have to let the customer know, the consumer know, the patient know that this is Blue Dream, but it was grown deviating from standard growing conditions. So if it's a little bit different than something that you are aware of that's grown under standard growing conditions, this is why. And I don't think that's a problem because like I do a Granny Smith apple analogy. We all know what a Granny Smith apple is. We have a certain level of expectation of what that apple will be when we purchase that from the store. However, if you have Granny Smith apples that were grown in California versus Tasmania, for example, there's going to be a level of variation there. And whether you know genetics or any of the science stuff, why. You just do.
Mm-hmm.
Grapes grown in France are going to be different than grapes grown in California. And why. Even if they started out with the exact same grape, you have a level of expectation of what, products from that grape will taste like, but it's going to be different and why. And you will have a preference as well. You can say, well, I don't like those French wines. They taste funny or whatever. I like my California Napa Valley wines. So I don't think it's a problem. And I also think that growers can put their name on it so that eventually once you start your cannabis journey, you can find out, where do you like your medicine or who is the grower that you prefer, how they do it. You know what I mean? And you can start making some informed decisions. When you start drinking beer in your teens or when you turn 21, you start with Budweiser because that's what everybody drinks, right? And then you start realizing, oh, there's a lot more to beer than just Budweiser. And you start kind of reaching, introducing your palate to some other beers. And then eventually you end up in the craft brewery and you're like, okay, I know this is a, New England IPA. Here's my expectation. Now who has the best one?
Right, right. Well, cannabis is definitely is on its way to being in that space because there's going to be, connoisseur and handcrafted brands and so forth.
Absolutely.
Why did my phone just answer? That's very strange. I had it turned off. It was very weird. Anyway, so yeah, that's definitely going to happen. I look forward to handcrafted cannabis because we're losing a lot in the certain areas, especially in medical, to where people are just, they unfortunately there's bad players in every state where they grow it, they dry it without curing it, and they get it on the shelves within a couple of weeks. And unfortunately, it's probably some of the type of samples that you're getting may not be cured as well as they could be. In your research, is there anything scientifically that you found out that surprised you?
The ChemDog thing was a big surprise. I honestly thought that they were going to be all over the board and it was just a thing. That was probably the biggest surprise. And that the National Institute on Drug Abuse has been growing research grade marijuana for at least for a while. The sample.
You talking about Ole Miss, that research marijuana?
Yeah.
Okay. Cannabis. Okay.
Yeah. So they're the sole provider of research grade marijuana for marijuana studies in the United States, appointed by the National Institute of Health. So, and NIDA, which is a little under the National Institute of Health. So the National Institute on Drug Abuse. So I managed to get my hands on some of their plant material because our chemistry lab was doing some work on THC and CBD. And it was, if I had have asked them for cannabis to look at the genetics of it, they would have absolutely said no. But and in the chemistry lab, I got to tell you, their material is locked down tight. It had to be in a freezer, in a fridge that was bolted to both the floor and the wall that has a lock on it, a coded lock. Then inside the fridge had to be another safe that also had a coded lock on it, and the plant material was kept in there. They had to account for every single piece of cannabis that went in and out of that fridge. Everything had to be written down and recorded, and the DEA would come in and do checks whenever they wanted. And so I couldn't take any of the plant material out of the lab, so I didn't, but I did go in there and I did extract some DNA, left the plant material in the lab, and then went back down to my lab with just the DNA. And so I was able to look at the DNA and how that compares to other samples that I had of cannabis that had wild collected hemp. I had cultivated hemp. I had some high CBD varieties. And then I had, indica sativa hybrid types from dispensaries. And genetically, this stuff from the University of Mississippi was more related to the hemp varieties than any of the dispensary varieties.
Was there a any terpene profile or the THC content, or couldn't get that?
Didn't look at any of that stuff. That's not the question I was asking. But no, it didn't smell like anything. And there was another paper that was put out that shows that so the thing about the research grade marijuana is that it needs to be, what's the word I want to use? It needs to be like, if you're ordering it for a medical study, it needs to be standard. Like it all needs to be 10%, or all needs to be 8%, or all needs to be 6%, depending on what you're trying to research. And they cut it. So when they grow it, they cut it with leaves and sticks to make sure that it's at that THC percent that they are giving for research. So I didn't want to know any of that stuff. I just wanted to know, genetically relatedness wise, where does it fall on what I call the genetic spectrum of cannabis? And I was very surprised to learn that it was most like hemp. And as was ruderalis, and nothing, not at all like anything that anybody can buy from a dispensary. So basically any studies that came out with, the effects of smoking marijuana that used this stuff from the National Institute of Health grown at the University of Mississippi, really what they were testing is hemp.
Wow, that's terrible considering they were giving it to 13 people monthly for 30 years, and there's only I think just Irv Rosenfeld is the only one left getting it now. He's in Florida, but I always heard that stuff was like really.
And it doesn't work. Well, no wonder it doesn't work, because it's not medicinal. What we would call medicinal grade cannabis. Right. It doesn't have anything in it that would be medicinal, barely at all.
So where did the research come from that that told us when U.S. Patent 6630507 was done by the United States, that it was good for shrinking tumors and Alzheimer's and Huntington's and all that stuff? Is that research from Israel or some other country, or they weren't getting it off?
I don't know. I haven't read, but yes, it could be from research from Israel, probably not using research grade marijuana from the National Institute of Health. But, something else that we came across is that the University of Mississippi hasn't harvested since 2014.
Wow.
Because they haven't needed to. They haven't had the demand for research grade marijuana, so they just haven't grown any. And I'm like, and I've seen the way they store it. They store it in these big cardboard drums, in plastic bags. It's not vacuum sealed. It's not. And I'm just like, what is that? What is that? So if you're ordering cannabis for your research, what you're getting at this point in time, what are we, 2001? You're getting 7-year-old like marijuana that was harvested 7 years ago. We also don't know. We know that when the program started, they had over 50 different strains. Now they don't have strains listed. You can buy very high THC. You can buy high THC. You can buy THC/CBD. You can buy high CBD and very high C. Like they don't even tell you what it is. It's broken down by THC and CBD, which we know there's more to cannabis than THC and CBD, right?
They haven't studied the terpenes yet, have they?
I mean, El Soli in his lab has, but it's not part of the NIDA program.
Right.
So that was problematic, and we don't know how they grow it. We're assuming, I am assuming that they grow it outside. we don't know if they're keeping seeds and pushing them through for the next harvest season. These plants could have been open pollinated for the last however many grow cycles, which would totally explain why they're more closely related to hemp than marijuana. But we don't know. We don't know why the stuff from NIDA is so different than anything you can purchase on the market today.
Well, they were given a mission, I think, Anna, to find out what's bad about cannabis. And that's what they were trying to find all those years, is what's bad about it.
Right. But I don't, it's a mess. But this was supposed to blow the doors off of this, and it is a monopoly, but it's also like a legal thing, part of the National Treaty of 1961. We have to have an appointed entity that can grow cannabis for research purposes, and that is the University of Mississippi. And yes, they are overseen by the National Institute of Health, who does have an agenda. Well, especially the National Institute on Drug Abuse has an agenda. But the researchers have been screaming, we don't want this shitty weed that you're giving us for research. Like, this is nothing like what my patients have access to. We want our patients to be, we want research done on something comparable, so that people can be confident that the research is actually reflective of what they will be able to purchase on the legal market.
And that's what Sue Cicely has been suing and fighting for all these years. And I think...
And the DEA came out a couple months ago again. They did this in 2016 as well. DEA came out again saying that they were going to open it up to applications to other institutions who want to grow research grade marijuana. In 2016, they took 35 applications. Those have been sitting on the DEA's desk since then. They haven't even looked at them. They just recently, in the last couple months, opened it up again, and I'm not holding my breath that they're going to actually.
I've actually heard that DEA license have been issued for research here in New Mexico, like at least 3 research facilities. One huge one by some company called Bright Green, out of.
Is it for research purposes, or are they just doing research on cannabis?
What's the difference? What's the difference between those two?
So for research grade marijuana, you're gonna apply to some sort of research grant through the NIH. So we are going to look at the effects of cannabis on Alzheimer's, and then they would send you your research grade cannabis, and you would conduct your study on Alzheimer's or whatever the case may be. There are lots of universities now that are doing cannabis research. Many of them are on hemp because that's legal. Some are doing it on cannabis, but it's not on people.
Right.
So that's the difference.
So I'm going to find those articles and I'll send them to you, because I've heard of at least 3 here in New Mexico. This little New Mexico is going to have some huge facilities growing supposedly millions of plants for research. So we'll see where that stuff goes, and they are DEA licenses. I know one company that has one for sure.
Send that to me. I'd love to be up to speed on this, because I haven't really looked either, but the last I knew that the DEA said, fine, we'll start taking.
Reluctantly. Well, so what do you think, once the scheduling, well, right now we have Marinol and Drebinol. I'm not sure how to pronounce that. They are like a Schedule 3, right?
We have Marinol, Dronabinol, Sativex. I mean, some of them are like generic names, and some of them are like the brand name, and I don't know the difference, but they're formulations of THC and or CBD for specific indications.
Right, for seizures and
so Epidiolex is for seizures. I think Marinol and Dronabinol are for pain and nausea with cancer patients. But they're for specific things, and you can't just get it. You gotta
have you ever looked at genetic markers in those synthetically made THC?
No, because they don't have a genome. Those are just molecules. They don't have any genetic.
So the molecule is the molecule. Got you.
So THC is a different molecule than DNA, right? So there wouldn't be any genetic marker in an isolate or an extraction. You have to have the plant material. You have to have cells to get that DNA from.
So, with the possibilities of federal legalization coming sooner than later, I believe, because of many reasons, how do you think that's going to help you personally? Are you going to be able to get your own license to do your own research and grow it yourself and sort of pin things down?
Research is incredibly expensive. I don't have a lab. All the research that I did was through through an academic institution. And I'm really not interested in starting my own company.
Okay.
That sounds just like and especially in cannabis. I mean, we all know people who have gone through the growing pains. Some of them make it, some of them don't. It's just, I'm not personally interested in that. Although if, somebody wanted to partner with me or wanted to conduct research and needed some sort of, consultant or something to build into that, I would definitely be down, because I feel like the research is just so far behind the industry. Lots of basic questions haven't even been answered about the plant, let alone, the complexity of, effects. And
Do you mean by everybody? I mean, do you think Meshulam doesn't have all the answers yet?
no, he's specifically interested in molecules. He's in the pharmaceutical world. He's interested in molecules and cannabinoids. He's not terribly interested, I don't think, in the plant. I mean, once you get that stuff out of the plant, those phytochemicals out of the plant, that's where the interest in most medicine is. I'm interested in the plant and whole plant derived products, so full extracts and things like that. Taking out individual isolated compounds for me is not that interesting. I mean, there are lots of people who are interested in that, but that's not really what I'm a plant biologist, not a pharmacologist.
Or a chemist.
Or a chemist. No.
Right. They keep finding these new ways of changing molecules into new cannabinoids, like the Delta 8 and the THCP, someone was showing the other day on Hash Church, and THCO acetate. I mean, will there be a place for those type of cannabinoids in the future when they're made properly and purged properly and clean?
I don't know. I don't have a crystal ball and I can't answer that question. Cannabinoids are very interesting molecules, and you mess with the little tails and the little things that hang off of them, the functional groups and whatnot. They start to act differently in the body and produce different effects. However, I will say that I feel like we are dangerously close to crossing the line of where we were at with spice and K2 and those sorts of synthetic cannabis products. I feel like if you can pretty readily convert, I'm not right now, because of the majority of the way it's done, I'm not really a big D8 person. Changing CBD from hemp into D8 and then calling it legal because it's hemp derived, I feel like that's kind of pushing it.
Yeah, well, it's definitely a gray area, and that's why people are taking advantage of that right now.
And the only reason that D8 is even interesting is because in those places you can't
say it. That's right. Prohibition has made D8 a thing, because
and now they even have D9 derived from hemp, so it's legal. And I am pretty sure that's not going to fly.
So you just mentioned spice and K2 as being synthetic cannabis. Now, neither one of those, as far as I know, are derived from any cannabis plant, right?
Cannabinoids.
They are from cannabinoids. Mm-hmm. Is it cannabinoids or cannabinoids? Which is it?
I say cannabinoids. But a lot of Americans say cannabinoids. But I feel like we don't say cannabis, we say cannabis.
Oh, but in Europe they say cannabis. all over Europe. I'm listening on Clubhouse and people coming in from different countries. It's they say cannabis, and in India they say
I've
cannabis.
never heard anybody say that before.
Oh, every
I
day.
say cannabis, so I say cannabinoid. But you know, whatever. There's a couple things that like it's not a cannabinoid, which I've heard people say.
The one that gets me the most is strand
Yeah,
when
that's
talking
wrong.
about strains, but strain isn't right either, is it?
It is. It is.
It
It
is.
absolutely is
Strain is
100%.
okay. Okay, I thought that strain wasn't acceptable and it should be cultivar, but
No, that came from a line in a paper, and I won't mention anyone's name. Somebody who's not a botanist who said that strain is not a recognized formal taxonomic uh, um, distinction, and it's not. It's not a formal taxonomic rank in plants, but it exists. We definitely use that term in botany, and it's to designate a group of genetically related individuals. Cultivar, however, okay, so I use the example of puppies. Cultivar, you have to do more work. You can't just cross two things, grow something up, pheno hunt, and then say this is my cultivar. There needs to be a lot more work before you get to cultivar status, and cultivar status does have a taxonomic rank. So if you have two mutt puppies, two mutt dogs, and you breed them, you're going to get a litter of a bunch of different kinds of puppies. You pick the best one from the litter, the pick of the litter. This is your new strain, right? You call it Charlie. Charlie's not a cultivar. It's not a breed. It does exist. Charlie is a thing. It's an entity. It's alive. It's gorgeous. But in order for Charlie to become a new breed, you have to backbreed and inbreed and breed out all those unwanted traits until you have a stable genetic line, like a beagle, for example, where when you breed two of these things together, all your puppies come out as beagles. Then you can go to the AKC and you can say, I have this new breed. I'm going to register it. And they go, yeah, totally. That's the difference between strain and cultivar. So
okay.
the majority of time, strain is actually 100% right.
Okay.
Most of the time, cultivar is not totally correct. And I don't care either way. I just want people to stop saying it's not a strain, it's a cultivar, because technically it's not a cultivar, it's a strain.
All right. So we're going to throw another wrench into here. What's a chemovar?
A chemovar is if you have a strain, all of the differences that come out chemotypically, those are all chemovars. So within Blue Dream, you might have 20 different chemovars, and every time you grow it in a different environment and it comes out with a different terpene profile, cannabinoid profile, whatever the case may be, that's a different chemovar. And I feel like that is absolutely asinine to split up. I mean, basically every plant that you have is going to be a chemovar. And the majority of everybody in the whole world is never going to be able to describe a plant by its chemovar. Like, it's just not practical.
Okay. Let me ask you about terpenes and their molecules that can be found in many plants. Okay, how do you feel about using other plant terpenes in cannabis products?
Why?
Well, why? I guess because some terpenes are found in such low levels that maybe and they do certain things to our bodies. From what I've been hearing and talking to people about, you could enhance, like they say, if you eat a mango before you smoke or do an edible, it enhances the effect. Do you know if there's any real scientificness to that? Scientificness, is that a name, a word?
Sure.
I don't know.
It sounds fine to me. Scientific validity. I'm not a fan of people adding terpenes to anything because, first of all, abundance doesn't equal potency. We don't know what a good amount of any kind of terpene to add to something is appropriate, especially when we're smoking things.
Yes.
A lot of terpenes are incredibly caustic. They are irritants.
Yes.
And when you light it on fire and inhale it, and your lung tissue is very sensitive, very soft, and you can damage it really easily. So when you're adding things to cannabis products, I personally think that's very dangerous, and I don't think people should be doing it. And not only that is terpenes work in concert with each other. You can have terpene A that smells like mangoes, terpene B that smells like hops. You put them together and they smell like chocolate. Like,
Is that true?
no,
Okay.
no.
Oh, okay.
And it could be you need exactly this amount of this one and exactly this amount of this one to get this kind of effect. We are awful at that kind of thing. And I personally know somebody who, like, this is his jam. Is he's a scent scientist, scent psychologist, and Chanel number 5 has never been able to be reproduced because they add something to it that no one else has put, that secret ingredient. They haven't put their finger on it, and it could be something as much as 0.1 of a micromil of ingredient X that nobody can figure out yet. So as far as terpenes go, like, I know there's a lot of different terpene formulations that you can purchase to make your this and that smell like ChemDog or Blue Dream or whatever the case may be. And I feel like inherently there's a problem with that, because what does ChemDog smell like? Because if you grow it here, there, and there, there's going to be 3 different sort of profiles.
So how long ago were you in Colorado?
I moved in at the beginning of April.
Oh, okay. So you've been there for, you were there for a bunch of years. So within the last 5 years, I interviewed a company in Colorado that was doing like vape pens with terpenes in it that they were testing with people. And there's videos on YouTube somewhere. I can't think of the name of the company, but they were, having people test and say, so how do you feel? Well, say, I feel happy. And then the next person, well, I feel a little drowsy. So there's different combinations of terpenes that can help the effect in cannabis or give you an effect. Do you remember hearing about any company in Colorado that was doing that? And
I mean, I don't know who it would be. I know there are some companies that do terpenolates, where it's like they call them terpene isolates. I think Extract Labs has a line of just terpenes. And there's a bunch of them, but yeah, I mean, why not? If you want a certain terpene profile, if you want it, I mean, there's so many different types of cannabis out there. There's so many different strains. I feel like you can definitely find what you're looking for. I don't think there's any reason to be enhancing anything without even knowing what you're doing, really. And nobody does, because nobody's done the science. It's just, we're talking about putting things in people's bodies, into people's lungs.
yes.
And I don't know. I worry about people who just are cool with that.
Yeah. Where do you see the research going in the next 5 years and 10 years? Being that you will be able to get some decent cannabis to be researching. I promise you that.
They considering the, how big the industry is, the economic importance of the industry, and how little we know about all of these things. I mean, if you want to get into cannabis science, do it. I mean, as soon as it becomes federally legal, it will be so much easier to do research. In Canada, they're ramping up their research because they can.
As far as going into that field that you're in, researching cannabis, is there currently a go-to institution to where they have good education happening, and where's the best place to get this education?
That's also a great question. I mean, relatively speaking, there really are no experts in cannabis, at least not teaching at university type institutions. There are a couple of programs that you can do, like, Amsterdam University, but it's not really a university type, you don't actually get a degree. And I'm not entirely sure, the qualifications of the folks that are teaching. Are they university professors? Are they chemists? Are they, what do they do? I don't know. I haven't really looked.
Right.
I know that the whole set of University of California, I think they all have cannabis programs now. But as far as like how much science you can get done, I don't know what all is out there. I teach modern cannabis science at CU Boulder, and it's an online class. Anybody can take it.
Excellent.
Yeah, and it's a good comprehensive class, but again, you don't walk out of there with, a certification or a degree in cannabis. And I don't know that there is any. I think there's at the University of Guelph. I think they're doing, and that's in Canada. They do some cannabis studies, cannabis research. The Institute of Cannabis Research in Pueblo, which is an offshoot of Colorado State University, you can do a degree in cannabis. You just have to look around. People are starting to do it, but again, because of the federal legality of it, more often than not, any university that's doing cannabis work is working on hemp.
Right. Physically, anyway. So when did you, before we go, I want to ask you two things. When did you first hear about the endocannabinoid system? And what do you think? Do you have any idea what you'll be doing in 5 years?
Those are both great questions. So endocannabinoid system, I really didn't hear about the endocannabinoid system until I was knee deep, waist deep into cannabis studies. I'm not a physiologist. I don't deal with humans. I'm a plant biologist. So as far as what happens outside the plant, it's not my area of interest or expertise. But I was curious because we didn't even find out about the endocannabinoid system until relatively recently. And there has been some, people have said, well, you don't hear about this stuff when you're taking anatomy and, in college. And I was like, well, I took anatomy in college. I took it, all through 1992 to 1996. And then I taught anatomy for multiple years from 2009 through 2019. But no, we don't. It's in our textbooks, but it takes up about a paragraph. And it's a whole system. It really should be it should have a chapter dedicated to it, just like the lymphatic system or the musculoskeletal
Well,
system.
that's definitely happening now. Nurses and doctors, they all need to be educated. Law enforcement needs to be educated. There's a lot of education that needs to be done. And I'm sure that, I know that there's a book out on the endocannabinoid system by Dr. Regina Nelson, who was in Colorado.
It's not in standard textbooks. It's not.
No, that's not. No, it isn't. But that's the beginning of people writing about the endocannabinoid system and learning about it. I just think it's fascinating with the receptors and all that stuff. And before we go, we have a couple minutes left. This is probably not in your purview necessarily, but cannabis hyperemesis syndrome, do you feel it's a toxicity of THC in certain people, or do you think it's chemically from what's being used grown on it?
No, it's definitely from cannabis. I don't know if it's from THC. It seems to be in people that have that consume heavily. But I don't think it's a chemically, it's not neem oil, it's not pesticides. And the reason that it's not is because neem oil has some similar side effects. Neem oil toxicity has some similar side effects or some similar symptoms, but there are also some differences. And the other thing that we know is that, people have grown without anything, which doesn't necessarily, because cannabis is a hyperaccumulators, so there could be something in the soil or whatever.
Right.
But the other thing is that even synthetic cannabinoids will set a person with CHS off. And those are synthetic cannabinoids. So they've never had any pesticides, neem oil anywhere near them. And for that reason, I do believe it is a toxicity issue. Something about the receptors just probably being overwhelmed. And it's definitely a medical condition.
Yeah, I have a friend that gets that every 18 months or so, and he just takes a few weeks off, and then he's back for another 18 months. So, Dr. Anna Schwabe, thank you so much. I'm sure we could talk for another 2 or 3 hours, but we'll do that. We'll do that again. We'll do it sometime again. Thank you so much for being here today and sharing your expertise, and I look forward to what you're going to be learning and finding out and telling us in the future.
Yeah, thank you so much for having me. I really enjoyed it.
My pleasure. We'll be right back. We'll be right back after this.
This is the Medical Marijuana Radio Show with Larry Love. All love.
I'd like to thank Dr. Cannabis. Thank you for all of your information. It was fantastic. I also want to thank my two radio mentors, both with the initials HS. And we'll be back next time with another great interview. Peace and love, everybody. Peace and love.